Hello, my name is Mark Dobson, and I am the coordinator of the MiCCuR project, Microbial Consortium for Enhanced Copper Recovery. And this transnational consortium is made up of 6 partners, myself here in Sweden, 3 partners from Germany, the University in Cape Town in South Africa, and PUC here in Chile. And we are made up of 5 universities and 1 SME from Germany, which is GEOS, and you can see the requested funding here. So consortium building, well, this consortium came out of a previous ERASYS app-funded project, which was called Sysmetex. And it was for optimizing acidophilic biofilm formation from metal sulphide dissolution. And it was made up of a number of partners like myself, Hans Garpowisch at RUP, Mario Vera, now at PUC in Chile, as part of the Sysmetex project. And this project generated some of the hypotheses that were then taken forward into this ERAMIN2 application, MiCCuR project, to be tested. And also additional partners were added because of their relevant expertise and the necessary abilities to be able to perform the ERAMIN project. So the work structure starts with chalcopyrite, which is a copper-containing mineral, in shake flasks that do the initial experiments that feed into bioreactors and column experiments. And this again feeds into a pilot bioheap for metal recovery and life cycle analysis. And the idea is that we improve the leaching efficiency and we use modeling of these different stages, the piloting in the pilot heap and process engineering to be able to develop a complete start to end process, including life cycle analysis and business analysis. So the key objectives are that we entered the project with 2 hypotheses, but both of them had the same end goal, and that is to control the redox potential during chalcopyrite bioleaching in a specific range, which has been shown to be the most efficient for releasing the copper from the chalcopyrite mineral. And these 2 hypotheses were to maintain a mixed culture of what we term weak iron oxidizers in the bioheap. And an alternative hypothesis was to inhibit the growth of strong iron oxidizers using chloride. So the start of the project was to generate laboratory-based data to inform on these 2 strategies, and then to feed into the column reactors and the modeling of the data that again informs on the optimal bioheap conditions. And then the main part of the project is to test the most promising of these 2 strategies in a pilot bioheap. So some of the results that have been generated so far in the project is that there's a published study on the investigation of signaling molecules between microbial species in the strong oxidizers to see if that can inform on how we can keep these strong oxidizers out of the bioheap and to stop them from forming biofilms. And a second manuscript is in preparation on species interaction, the effects on signaling molecules and the redox potential during bioleaching. And then work on the weak iron oxidizers is that a large number of species, as you can see here, have been tested for their bioleaching capacity and also for maintaining the redox potential in the desired range and therefore having efficient copper bioleaching. From these single species experiments, we designed mixed consortia that were tested at different temperature ranges that were relevant for the bioheap. And all this data was fed to our partner at UCT in South Africa for modeling and the column experiments. And also proteins have been sent to RUB in Germany for a proteomic analysis, so we can understand how the consortium is working and the biology behind it, and how we can hopefully control the bioheap. Further work has been done on hypothesis 2 using chloride tolerance to keep the strong iron oxiders out of the bioheap. So then in a pilot bioheap, the water that would be used would have some chloride in it, and this inhibits the strong oxidizers, hopefully keeps the weak oxidizers dominant. We can control the bioheap. And again, data is being sent to UCT for modeling, and the column experiments and proteins will be sent to RUB for analysis. So then these are the column reactors that are being run at UCT in South Africa. We have the modeling of the bioleaching in a continuous flow reactor. This has been performed. We're calibrating the model with the data from the column experiments, and these weak ion oxidizers and the sodium chloride experiments are presently underway. And this, again, will give us stronger data, more realistic data that will be fed into the bioheaps. And what you can see here is the bioheap pad being constructed in Chile, and microbial culturing is underway for the inoculum to be able to grow up to inoculate our bioheaps. And the planned bioheap start is in March 2022, and this will be in operation, we hope, for up to 12 months. And this is the main part of the project. This is the main goal. This is testing the hypotheses in a pilot scale to be able to give us realistic data on the ability of our systems to be able to efficiently extract copper from chalcopyrite. In conjunction with all this scientific and laboratory experiments and the pilot, we are running a comparison of the MiCCuR concept to conventional copper extraction techniques such as smelting, and we can do inventory analysis, life cycle, interpretation, and conclusions to be able to create conclusions and recommendations on the most efficient and also the best environmentally sound practice for able to be extracting the copper from the chalcopyrite. So the expected outcomes from the project is data on heap leaching of the chalcopyrite at low redox potential, to create a flow sheet from start to finish of the chalcopyrite bioleaching process, including recycling of liquors and the copper recovery, life cycle analysis of the complete process chain in comparison to traditional smelting of copper, and the technical and economic evaluation of the process. So how has ERAMIN benefited the project? Well, this project is aimed at topic 3, the processing, production, and remanufacturing. It is aimed at improving the efficiency of raw material supply within the EU, and this chalcopyrite is a low-grade deposit. And the subtopics within the call that we're addressing is bioleaching, reduction of waste, as this is a biological process, energy efficiency, because it doesn't involve smelting, which is a high energy, high temperature process, and bioleaching has a lower environmental impact. And the way that ERAMIN has benefited us is that we've been able to include world experts. We've been able to include partners from Chile who are able to have this pilot scale bioheap. And with that, I'd just like to acknowledge ERAMIN 2 and the European Union, as well as thanking MiCCuR in Chile, who are providing the low-grade copper ore that we will be testing in the bioheap. Thank you very much.